Welcome back. In this video, we will discuss about the NAT problems related to biology as they appeared in the IIT JAM Biotechnology examination in 2021. Let's start with the problem number 41. Here, 1.45 gram of sucrose is dissolved in 30 ml of water and you have to calculate the molarity of the resulting solution. First, I will calculate the molar weight of the sucrose in this way. Here carbon is 12, hydrogen is 1 and oxygen is 16. So molecular weight of sucrose is 342. Now, according to the definition of the molarity, if 342 grams sucrose is dissolved in 1000 ml water, it is called 1 molar sucrose solution. So 1.45 grams sucrose if dissolved in 30 ml, the molarity of the solution will be 0.141 molar. And in this problem, the maximum number of allele present on human chromosome 4 is 2. So you have to calculate the total number of allele present in 5 male and 10 females. We know that chromosome number 4 is an autosomal chromosome. So whether the person is male or female, it doesn't matter. It will be diploid or 2 copies will be present. So total number of individuals are 10 plus 5 or 15 here. So the number of total allele will be double, that is 15 into 2, that is 30. Now in the next problem, you have to calculate the total number of peptide bonds present in a 20 residue long linear peptide. We know that in order to link the first 2 amino acids, only one peptide bond is formed. Next, for each amino acid, one peptide bond is required. So we can say that total number of peptide bond will be n minus 1. Where n is the number of amino acid that is present in the peptide. So in this problem, the total number of peptide bond in a 20 residue long polypeptide will be 20 minus 1 or 19. Here the molecular weight of the human IgG light chain is 24 kd and you have to calculate the total molecular weight of human IgG. The molecular weight of light chain is 24 kd, so the molecular weight of heavy chain will be 48 kd. And in human IgG, we know that total number of light chains are 2 and total number of heavy chains are also 2. So the total molecular weight will be 144 kilodalton. Here, you have to calculate the maximum number of genotypes possible for the gametes formed in a diploid cell. The genotype is provided here. The maximum number of possible genotypes for gametes can be calculated by this formula, that is 2 to the power n. Where n is the number of genotype. Here n is 4. So 2 to the power 4 is 8. Now in this problem, at 25 degrees centigrade and pH 7, the concentration of glucose 1-phosphate and glucose 6-phosphates are provided. The standard free energy change for the conversion of glucose 1-phosphate to glucose 6-phosphate you have to calculate, and the value of R is provided here. We know that equilibrium constant K equilibrium is the concentration of product divided by concentration of the substrate, that is 19. So I will calculate the temperature in Kelvin first. Here 25 degrees centigrade means 298.15. The standard free energy change can be calculated using this equation. Here the value of R is provided and the T we have calculated here. And the value of K equilibrium is also we have calculated. So the standard free energy change is minus 7298.5 joule per mole. The number of polypeptide chain in a core nucleosome you have to calculate. We know that a nucleosome is consist of a DNA and a protein that is called histone. And the core histone contains 2 copies of each, H2A, H2B, H3 and H4 subunits to form a octamer. So here in a one nucleosome, the total number of polypeptide chain will be 8. Here the researcher forgot to add one primer and he completed 25 cycle of PCR. Now you have to calculate the how many copies of DNA will produce. You can check this picture. If only one primer is added into the master mix, at the end of each cycle, this complementary strand will amplify. But this strand is remain same. So after 25 cycle of PCR reaction, the amount of single stranded DNA produced is 25 copies. A double helical DNA molecule is composed of 32 mole percentage of adenine. the mole percentage of cytosine in this DNA molecule you have to calculate. We will use the Chargaff's rule to calculate the percentage of all 4 nucleotides. If A is 32 mole percentage, that means thymine is also 32 mole percentage. The total is 64, and out of 100, 36 is G plus C, or cytosine will be 18 mole percentage. In a compound microscope, the magnification power of objective lens and eyepiece are provided. You have to calculate the total magnification power. We know that the total magnification power is the magnification power of objective lens multiplied by the magnification power of eyepiece, and here it is 1000x. And in the last problem, in a population of Hardy-Weinberg equilibrium, for gene X, only 2 alleles, namely capital A and small a, are found, and if the frequency of allele capital A is 0.2 and the frequency of allele small a is 0.8, then the frequency of heterozygote genotype capital A small a in that population will be, and you have to calculate up to 2 decimal places. We know that according to Hardy-Weinberg law, the genotype frequencies and allele frequencies are in equilibrium if the following assumptions are met. According to Hardy-Weinberg equilibrium, we will get this relationship, and here capital A and small a are 2 alleles and their frequencies are 0.2 and 0.8, and we need to calculate the frequency of 2 into capital A into small a, and that is 0.32. So that's all for now. I hope you like this video, so don't forget to press the like button, and if you are watching my video for the first time, then press the subscribe button and the bell icon, and lastly, thank you all.